So Dr. Daryl Scott, he's been, how many conferences have you been to now? Is this 6 or 7? Quite a few. So there's so many new faces here that a lot of you have not heard him talk before or met him. So he'll be part of the panel this afternoon to be able to answer questions. He has a really great way of explaining things, things that are really complicated and go over your head. He's able to kind of bring it down a little bit, which is great. And I also, I want to give a really big shout out to his family, and as he likes to refer to, his cute wife, April. They have been helping with childcare. He lives locally in the Houston area, and obviously so his family does as well, and they've been helping out downstairs. So yes, thank you. Yeah. It was very generous of them to be sharing their time. And they were here yesterday, and he was still at work. So anyway, here's Dr. Scott.
That's fair. So this is really loud up here, but that's a good volume then back there? Yes, hopefully? Okay, so I was, well, I have been to several different conferences, and I love being here, and I love bringing my family. And I tell people that we'd like to go to these conferences because my family doesn't always get a chance to come to clinic to see the kids that I work with. And so when they come here, they get a better feel for just the things that I do and the awesome families that I get to deal with and visit with and help out. And so it's been a great thing. We've gone to, again, many different conferences. And I know many of you, but I also heard that there's a whole lot of people who are brand new. I also know that when you hear something once, you catch a little bit, and then you catch a little bit another time, and sometimes for third time, fourth time around, you finally get it. Anyway, I hope that this presentation will be helpful. But I want to first just kind of introduce myself. So I have multiple roles, as all of us do. And so I'm a parent. So I have 7 kids, and life is busy. I often get asked if I have a child that has 1p36, and I say no. But we deal with our own genetic problems. There's very few families that actually don't. And so this is one thing. Our family deals with other things. And if you talk to people and you get to know them, usually there's some things in almost everybody because we are all imperfect. And so we all deal with our own issues. I'm also a doctor, so I'm a clinician. I'm a clinical geneticist. So I don't know if any of you are my patients. Yes? That's good. So I see patients here at Texas Children's Hospital. And I'm also a scientist. And so I have my own research lab. I mostly work with mice in my research lab, but we also do a lot of human studies. And again, we're located just not very far from here in Texas Medical Center. And then we work with a whole bunch of different hospitals in that center. And so 1p36 is actually one of the main places that works on 1p36, even before I came here, was Baylor College of Medicine. And so a lot of the research that's been done has been done right here. So one of the reasons I think it's worthwhile, or one question to ask that's worthwhile asking is, why study 1p36? I think there's a lot of reasons why we do so. And each one of the roles that we play may make us look in a different way at things. So the parent side, that's most of you, and that's why you're here at the conference, is because you want the very best for your children. I want the same thing. I want the best for the children I see in clinic. And there's certainly a lot of doctors who have interest in 1p36. It's super common as far as genetic disorders go. And so for us, 1 out of 4,000, that's a common thing. So as a clinical geneticist, I should know this. And so if you're a geneticist should say, yes, I recognize this, or I can very quickly put my hands on information about that. Your pediatrician, however, probably won't. And so a typical pediatrician will see between 1,500 and 2,000 kids. And so if you consider that 1 out of every 4,000 has 1p36, you are the one patient that they have. And so very rarely would you have a pediatrician who sees more than one child that has 1p36 deletion. At least that's been diagnosed. There's also a whole lot of more children that probably should be diagnosed but aren't. And some of you have dealt with that. We sit and we talk and say, yeah, my child was diagnosed when they were 5 or 6 or 7. It's just that again, the ability to do these kind of tests are there, but often doctors are unaware of how to do them or how to make that happen. We also recognize that your kids have a lot of different medical problems. And so we're interested in trying to solve those or prevent those as best we can. Then there's the scientist part. I must admit, I'm a scientist and I kind of like this part. But at the same time, I would say I'm driven more by the doctor part of anything. But scientists all over the world are interested in learning about genes and learning how those genes affect development. And so even people who have never heard of 1p36, they're actually working on this disease. They may not even know that they're working on it, but they are. And that's because they have interest in how things develop. So many of your children probably have heart defects. Some of them have seizures. Almost all of them will have some kind of learning issues. And so there are scientists that are working on all those different things to try to understand why are those genes and what are the genes that are important in making heart work or heart develop or brain function. All those things are being researched. So there are some things that we all kind of want. And this is the kind of things that I like to focus on when I do my research. And so all of us want to know what problems to expect. So one of the things that often comes, or when families first come, or the diagnosis is first made, often it's made when a child is very small. And sometimes it can be just because of one issue that's cropped up, and parents want to know what are all the other issues that we might face. I imagine you've questioned that same thing of your doctors. So we all want to know what problems to expect for each child, and also how to prevent or treat those problems. We also realize that there's some challenges that we face in 1p36. I'm a clinical geneticist, and so I'm supposed to be the expert in genetic disorders. When I first started, there were about between probably 3,000 and 4,000 disorders that were out there that were genetic disorders. A clinical geneticist will usually know maybe on the order of 200 or 300 by heart. In other words, I can walk into a room and say, that child has this syndrome, and I know about it. For all the other thousands, we have to use the internet and try to figure things out, just like you might use the internet to figure out a problem. We are specialists in things that are so broad that we have a very hard time being specialists in these things. 1p36 deletions are even more complex because this is not a single diagnosis. This is a collage of all kinds of different kids who have different genetic problems. And so no two children here are probably the same, not genetically. In fact, it would be extremely rare to have two kids that would be exactly the same unless they were siblings. Why is that? So if we just take a look at 1p36, that's that top part of chromosome 1. Chromosome 1 is huge, and so it's the biggest chromosome, and so this is only the tip of it. And that tip takes in about 30 million letters of DNA. And so when we say, well, my child has a 1p36 deletion, it could be that they're deleted for this much. That would take in 99 genes. Or they could be deleted for this much. That'd be 186 genes, so twice as many genes. You can imagine that the medical problems that a child with a small deletion has may be different than a child with a big deletion. That's complex enough, but there's also kids who have deletions that come in the middle. So you can also make a middle deletion or an interstitial deletion that might be 77 genes. Those genes may actually be quite different than the genes that are deleted in this child because they're not even the same part of chromosome 1. And so literally when we come to a conference like this, yes, we all have the same deletion or same syndrome, but at the same time, when you really get down to it, none of us have the same syndrome. And so that makes it a little difficult for us as doctors because I have to figure out what is the piece that is missing in your child. And what does that piece do? And so, again, this is more complex than most of the diseases that we work with. So in addition to not having any two kids be exactly alike genetically, we also don't know a whole lot about the genes that make a difference. In other words, we're still learning about genes on 1p36, and I can't put my finger necessarily on all the ones that cause the heart defect or cause the brain defect. because we just don't know what a lot of the genes do. There are about 20,000 genes that are in our bodies or in every cell. Those 20,000 genes make proteins that then make the rest of our body. Only a portion of them are on 1p36, but there will be genes for every piece of your body on there. So there's eye genes and heart genes and brain genes. We know that because of all the different problems that kids with 1p36 deletions have. And so there must be genes that control all of those things. The question is, which ones are they? And what exactly are they doing? And how do they work together? That's a big challenge. To try to kind of illustrate this a little bit better, I was thinking about this. I've taught this I don't know how many times, and every time I try to think about a different way of doing this, I want to try something today. I want to compare 1p36 to a piano. How many of you actually play piano? A few of you. I'll just tell you that I have played a keyboard in the past, not very well. The reason why I played was because I'm a member of a church, and we have a lay ministry, and my dad was the pastor. And we were living in a small, small island off the coast of Malaysia. There weren't a whole lot of members of our church in that little island, and the one person who could play piano was about to leave. So my dad came to me and said, "Son, you have 6 months to learn how to play the piano for church." I was like, "What?" I said, "We don't even own a piano." And he says, "We will buy one tomorrow." And so we bought a keyboard, and I started to learn how to play piano. And the only thing I was supposed to do was figure out how to play hymns. And so I learned how to play hymns. But I'm not a very great piano player. And so if I make mistakes in the way I describe things, you just give me a little bit of slack, okay? Because if you're a better piano player than me, that wouldn't be very hard. But let's talk about pianos. I know that pianos have keys. They have white keys and they have black keys. I also know that there's some special things about keys. And one of the things that I know is that there's a thing called a pentatonic scale. Anybody heard of that? Whoa, see, this is what's scary. People actually know these things. If I've got this correctly, a pentatonic scale, or a song that's written in pentatonic scale, can be played with just black keys. I'll say that one more time. On the pentatonic scale, you can play the song with just the black keys on the piano. The white keys don't matter or aren't needed. And so the white keys aren't needed, but you have to have the black keys. If you were to do something really rude to your piano student, you could give him a piano that has all white keys. Now you would probably say, well, there's some that are a little higher, a little narrower than other ones. But if you just said, hey, I'm going to give you this piano, and I want you to figure out what keys are important for playing this song. They're all white. How would he ultimately, or he or she ultimately, be able to figure out which ones are the black keys, the keys that are important? The way you would do that, or one way to do it, would be to take a bunch of pentatonic songs and then play them on the piano. And over time, you would figure out that, wow, yeah, like all the small narrow keys that are kind of higher up, they're the ones I'm using, and I'm not using any of the other ones. And so you could figure it out by playing enough songs. You could also do that by taking off and saying, I won't play that key, and then you see if it's actually used. Because the song won't sound right if there's a key that's missing, right? And so little bit by bit, a person playing an all-white keyboard could figure out, well, yeah, those are special keys. Those are black keys. And you could figure that out along with, again, you played enough songs. Now, thinking about keys like genes on 1p36, there are some genes that are just not needed. In other words, you can take out one copy of that gene, you'll still have another copy left over, but losing that one copy is not a problem. It doesn't cause any medical problems, and you could do just as well without that copy. In most cases, that's because you have another copy that's working. But there are some genes where you can completely lose them, and we don't know why. Like, we don't know why God made those genes. But you can lose both copies and be okay. There are, however, some genes that are absolutely needed. And when you miss those, even one copy, there are often consequences for that. Or the body has to find a way to get around missing that key in order to make things work correctly. And so just like in keyboards, there are black and white keys. There are needed and unneeded genes on 1p36. So if you were to say, well, what if I don't know what those genes are? How would I figure that out? Like, how do I know which genes are important? Well, one of the ways to do that would be to study people who are missing one gene, or they have a gene that doesn't work. Remember how we said we could play the pentatonic songs and figure out if we were missing a key that was important, the song wouldn't sound right? You can do the same thing by studying people that have a gene that's missing. How do we actually do that? What's really cool is in the last probably 5 or 6 years, we can do this very well, at least better than we ever could. One of the ways of doing this is to take a whole bunch of, and I put this in quotes, normal individuals, and actually check the genetic code of all 20,000 genes in them. Now this seems like a kind of maybe a silly thing to do. It's like, well, why would you want to look at the genes of a bunch of normal people? But because they are classified as normal, they don't have major medical problems. Now you can look at me and say, well, so Dr. Scott, are you normal? And the answer is, depends on who you ask, right? I've had 5 surgeries in my life. There are things about my body that are not normal, but I have a job, I have a family, and most people would classify me as normal. Even though, again, if you looked at my medical details, you'd say, well, hey, he's got issues, right? That's kind of the same thing that we've done here. Not we, but as a scientific community, there's about 65,000 people that are normal that have been sequenced. The number is actually even larger now, but they're all in a database. And then you can say, well, if I look at the sequence or the letter codes of all their genes, I can probably figure out which genes they have that aren't working because they have messed up letter codes, and which genes are working. This gives us a really powerful resource to try to figure out what individual genes do, or at least whether they're important or not for a normal person. So for example, let's say we have a gene that we're interested in. We could look at the size of the gene, and if it's a big gene, it will probably have more chances to be knocked out or hurt than if it's a small gene. And so we can actually predict whether a gene will have how many people by chance would have that gene knocked out or hurt. Let's say, for example, that we do that calculation, and for our gene of interest, there's 12. So we expect that in this group, if this gene does nothing, we'll find 12 people that have the gene knocked out or hurt. If we then can actually go back and look at the data and we find that 10 people have that, well, 12 and 10 are about the same. I mean, nothing's perfect, right? I mean, this is science, but these are biological people, right? But if you say 10 and 12, you'd say, well, that's about the same. So chances are this gene is not important. So far, so good? Yeah, so if these 2 circles are about the same, this is not an important gene. If, on the other hand, we were only to find 1, then you have our complete attention. Because we should have 10 times as many people in our normal group that have that gene. And yet we only find 1 that's missing that. And that 1 person could have a heart defect and just kind of snickled into our normal group, right? Just like I could be normal, but I'm not really. And so that would trigger us to say that's an important gene. And so by doing this, we can actually start to say, well, what are the genes on 1p36 that are important? And we just look at them by studying a bunch of normal people. How does that actually look? You could do this too. If you want to be a scientist, it's actually free. You can just go on and you can Google this thing. In fact, if you had a computer right now, you could go to this website. It's called the Exact Browser. And this is the thing that contains all the data for 65,000 people or so. You don't really have to do this, by the way. But if you want to, you can. And you can take any gene that you want and stick it in here. And it'll tell you how many of those 65,000 people have that gene hurt. So let's just do it. Let's put in a gene called TTLL10. Anybody heard of that? Yeah, me neither, as it turns out. Like there's 20,000 of these things, right? All of them have names. Most of them have little letter abbreviations, right? But I don't even know those. I know the ones that are important to me. But if you said, what does TTLL10 mean? If you asked 100 geneticists, there would be none that would know what that gene does. So, but let's put it in. So you put that gene in, and when you do, it makes this cool little graph of the gene. This is actually all the little sequence peaks from the different patients. So you can see that some parts of the gene are checked a little harder than other parts. But what we really want to concentrate in is this little box up here. And in fact, one line of that box. And one line of that box tells you how many people we expect to have that gene not work and how many people we actually find that don't have that gene working. And so in this particular case, the program predicts that 9.6, you can't really have 9.6 people, by the way, but the program says we should get about 9.6 people that don't have this gene working in this group, and we find 8. Now, this is the test. Does that mean this gene is important or not important? Not important, because the two numbers are quite similar, right? And so in this particular case, we'd say no. TTLL10, you could probably delete a piece of that. You could take that gene out, one copy, and we would probably see nothing. So far, so good? Okay, now we can give that a score. Scientists like scores because it lets you put them into big Excel spreadsheets, and then you can scan your genes and do all kinds of stuff. If a gene is not very important, we give it a score of 0. So when I see a 0 gene, this is not important to me. I'm looking for something more, something different. They call it a PLI score. It has to do with the probability of loss of function. You don't have to know that. But a PLI score to me means something. And so when I sit down and I talk with my other doctors, we talk about PLI scores. And I say, this is not important. Let's take a look at a different gene. This is a gene called PRDM16. How many of you know this gene? Ooh, a couple people. So some people actually know this gene. I know this gene. You may want to know this gene. But let's put it in. So you take PRDM16, it's a 1p36 gene. So is that TTLL10 too. In this case though, there's 33.5 people are supposed to have that, but only 2 do. Does this have my complete attention? Yeah. This is the kind of difference that says, Dr. Scott, wake up. Like, think about this. This is a gene that does not like to be deleted. If this is in your patient, it's doing something. And so this would be a very important gene, and it would get a PLI score of 1 because the difference is so dramatic. And so if I have a PLI score 1 gene, I'm automatically, as a geneticist, saying, I want to know what that thing does and what it might do in my patient. So far, so good? You can do this, right? So you guys never knew that you could be like clinical geneticists. This is just not that hard, right? You could do this at home. This is awesome. This is literally what we do in clinic. Like, I sit down and I do this kind of stuff with my trainees. Okay, let's take another gene. This is called KCNAB2. Anybody heard of that one? No, it's important to me though because this gene is important in brain and might be a gene that causes seizures in 1p36. And so I'm interested in this gene. It has not been proven yet, but I'll tell you why I'm interested in it. If you look at its score, about 14.5 people should have this, but only 2 do. The fact that 2 do may mean that this doesn't always cause a problem, but it's probably important in doing some things. And so its PLI score is 0.84, somewhere between 0 and 1, but it's still high enough that you have my attention. I'm just going to try to have to figure out how this gene sometimes makes a problem and sometimes doesn't. Maybe this gene works with some other gene, and when you delete both of them, then you get a problem. And so anyway, this is an in-between score. I have to deal with that, and I have to think about that as a scientist. Easier to just do 0s and 1s, right? But life's never easy. Okay, so how many genes how many genes on 1p36 score high? So if you just said, well, all the genes on 1p36 that I can score, like that system works, there's about 345 genes I can score on 1p36. Of those, about 49 or 14% have PLI scores that are greater than 0.9. That's a lot of numbers, but it's just saying that there's only a portion of those genes are going to be super important. If you look at it like a keyboard, and the black keys are important, this is what a piece of 1p36 would look like. This keyboard has 52 keys. It would be about the same as having a little over 4 megabases or 4 million letters missing. A lot of your kids will have deletions about that size. So it's a good, helpful thing. And you can look that actually there's a lot more white keys than black keys. In this particular case, probably about 7 or 8 of those keys are going to be important. If you say, well, that's only if it's greater than 0.9. What if you did like 0.8? Well, then you'd have a few more keys. In this case, there'd be maybe 61 genes over the whole 1p36. And in this particular section, there's probably a couple more keys, maybe on the order of 10 or 11. But again, there's a lot more white keys than black keys. And I can ignore the white keys because they are not so strong or so important. But I want to know what the black keys do. So far, so good? You guys are like, hey, I should open up a clinic, right? This is awesome. Like, I'm doing great. Okay, but so far, so good. One of the things you have to remember is that each one of your children is different. So just remembering with that visual, this could be your child because it may be a smaller deletion, right? And so your child's keyboard might look like this. And maybe there's only 4 genes that are really, really important. Or your child's keyboard could look like this, in which case, well, there's 5 genes that are important. This is the challenge. Now you're kind of, hopefully you're going, yeah, that's why so-and-so's child doesn't look like my child. Or that's why so-and-so's child can do this and mine can't. Or that's why we're having this problem and they don't. This is part of the problem, or part of the complexity of this. And we would like to be able to sit down with you and say, this is what your child is dealing with or will deal with. That would be the ideal, because everybody's going to be different. Okay, so do you have the information already in hand to know what your child's keyboard looks like? The answer is almost all of you do, but some of you don't even know that you have it. You have actually something that's very powerful in the hands of the right geneticist to be able to solve some of these issues. Most of your children will have been diagnosed using a chromosome microarray analysis. If they have not, they would have been used an older technique, but almost everybody now is done this way. So if your child's been diagnosed probably within the last 10 years, chances are one of these exists for your child. The chromosome microarray analysis, I'll just tell you how it works, because sometimes I have to explain this to geneticists, and I'm like, how'd you get through med school without knowing this? And so I don't expect you to know it either. But what we do is we take a sample from your child. We're interested, usually it's a blood sample, we're interested in actually the white blood cells, because white blood cells have DNA. Red blood cells, as it turns out, kicks out their DNA, and they're just kind of little bags of chemicals, but they don't have any DNA in them. So we take out the white blood cells, and we get DNA out of that. Then we use what's called a microarray, which literally is a glass slide about this big, just a piece of glass. But on that piece of glass, they've actually used a printer, like an inkjet printer, to grow DNA on the slide. If you don't think that's cool, man, you just have no imagination. Because that's like amazing, you can do this, right? But by using the special chemicals, they actually will grow different strands of DNA on the slide. Each one of those strands of DNA acts as a probe. And I'll explain kind of how this works. This is 1p36 again, right? So what it's going to do is on that glass slide, it's going to have little pieces of DNA from all over the genome. In other words, not just 1p36, but all the chromosomes. And those different probes, you could just like line them up, but every probe has a special place that it's from. In other words, its sequence matches a piece of DNA in your cells. There's actually far more probes on 1p36 than I'm showing, but like it would be so dense, it would be one big red line. So anyway, each one of those probes tells us, or can be used to check how many copies of the DNA are there at that spot. We then use this thing called a laser scanner. So we use the microarray, we put your child's DNA on that microarray, and then we stick it in this laser scanner. That laser scanner is worth way more than my house. Like, it's a lot of money to buy one of these. And so there's only certain laboratories that would have a laser scanner, and they're the kinds that would do this kind of test. So if you say, well, why can't I get that genetic test done at my local hospital? The answer is they don't own one of these, because this is extremely expensive and very special piece, so there's only some laboratories in the country that will have them. But that laser scanner will scan that piece of glass. In this case, this is an actual photo of a piece of glass, that's microarray that's been scanned by a laser scanner. This particular one has 1 million spots of DNA. It just looks like a bunch of yellow, right? But if you blow it up, it actually has different circles. And each circle is a piece or a specific DNA that's been grown on that slide. Again, if you don't think this is cool, man, you need to have more imagination. This is amazing, right? Little tiny dots. Now you can say, those dots look like they have different colors. That's actually important. So the way the laser scanner works is it will be yellow, either yellow, green, or red. And you can pick out those dots by looking at the array. And if I just erase them so you can see them better, if there's a green dot, that means that this child has one copy of that piece of DNA. That's too low. You're supposed to have 2 copies, one from mom, one from dad. If it has a yellow copy, that's normal. Or if it has a red copy, that's too much. Like there's an extra piece of DNA. And extras or less can throw things off. So both of them are bad. You want 2 copies. Now, you can imagine if I just gave you one of these slides and said, take this home and tell me what it means. You'd be like, what? There's like 1 million spots on that thing, right? So we use computers to help us to put all the spots in order on the chromosomes, and we get something that looks like this. Now, this is black, this is green, and you can see this, right? This is a deletion. I actually used to do this at home. It's kind of funny. I used to have to do these by hand. Like I have to go through them by hand and look at them. And my kids would sit around and like, hey dad, what are you doing? I'm like, I'm analyzing microarray data. And they're like, I want to do it too. I'm like, okay. So they would sit down by me and we would scroll through all the pieces of the chromosomes and they'd say, dude, dad, that's like a deletion right there, isn't it? I'm like, yeah. And like, we got to write that one down. How about that one? That's a duplication, dad. And they could do this. I thought it was kind of cool to do with my kids, but then they went to college and they would sit in their biology class and they would show these things and they'd be like, hey, dude, that's a deletion, right? And their teachers would say things like, so does anybody want to know what this is? And my kids would raise their hand and they're like, yeah. And they're like, well, it looks like you have a deletion on chromosome 1. Maybe it's on the Q arm. And they're like, what in the world? And the professors, their eyes would be like this big. And they're like, so why do you know that? And they're like, oh, my dad's a clinical geneticist. And they're like, ah. And they get like bonus points for that day. And this happened over and over again with multiple kids. I'm like, this is amazing. Like, anyway, you could do it too. It's not that amazing. But when you see this, the power of it is that you can actually take that, blow it up so you can see it better. And you can see all the little genes, right? Like this is where all the genes are. All these ones are different genes. And you can actually block it out and say, that's the piece that's missing. Not just like kind of that's the piece that's missing. That's the exact piece. And that's powerful because for any child, I can tell you what genes are deleted and what are not. I can tell you what their keyboard looks like. And then we can see what we can do from there. And you probably hold one of these, or your doctor does. What does it look like? It looks like this. This is a report from Baylor, which is where I work. But this is actually a report from a patient that was seen there. And you can actually look at this little piece, and it tells you the numbers or the actual letters where the deletion begins and ends. And by doing that, I can then go back and I can actually pull down all the genes that are in that section. Now, if there's a whole bunch, that's hard, but there's some kids that have a very small change, and it's very easy to actually list them out. But this is the keyboard, and there's a different one for every child. And that's powerful, or could be. So how do we use this information? Let's go back to this for a second. Let's say this is your child's keyboard, and you have those black keys that you're interested in now because we've been talking. You say, so Dr. Scott, what does those black keys, what do they mean? The difficulty is, again, I don't know what all the genes do. So the first thing that we used to do is we'd say, well, can we map onto that big keyboard of 1p36? Can we instead look at everybody's small keyboard and decide where different genes are working? I'll give you for instance. I don't know if you know this, but on 1p36, there's 2 special regions. They're called critical regions. If your child is deleted for a critical region, they could have anything that 1p36 kids have. Doesn't mean they will, but they are at risk for those things. And so we've marked out these critical regions so that we can be helped in that we know that those regions contain really important genes. There's 2 regions that we've mapped. One is called the distal region, which is just on the very tip, and one's the proximal region. The kids look different. The kids look different. Your kids might look different. So if you have a distal deletion, they may have straight eyebrows, deep set eyes, different characteristics, but you can also just look around and say, yeah, my child looks like that child. They have something in common. When you go to the proximal region, if you're missing just that, they don't look the same. They may have all the same medical problems, but their features are different. I'll just tell you, it's a lot easier to figure out a child that has a distal one than a proximal one because they look, this is considered the classic look of a child with 1p36. If your child does not fit that, do not cry, because that's okay. We know about other, like you're allowed to have a different keyboard, right? But looking at the keyboards, we can start to say, well, what piece of that keyboard is important? By checking the keyboards of kids and then seeing what their features are, we can know a few things. One thing that you probably never worried about, but your physician might have, is that some kids with 1p36 have large fontanelles. So when they're born, they have a big soft spot. It's a key. It's like one of the things we look for. There's actually a section or a gene or a piece for that, and that's this piece right here. We know that the gene that causes this was sitting right here. We've actually identified that gene now, so we know the exact spot. But back in the old days, we just knew it was in this section. There's also a section for hypothyroidism. If your child has that piece deleted, we should be checking them on a regular basis for low thyroid, which is it's basically a hormone that helps you grow and helps also your body to function right. If this is low, we can make your child feel better and grow better by giving them thyroid hormone. So we should check that. That gene has not been identified, but it's somewhere in that section. There's also genes for cleft palate and cleft lip and palate. Less common, but we know where it sits. We've got an idea of where some of those genes are, but not 100%. There's 3 different sections that are for seizures in that distal region. We know some of those genes, probably, but again, we are still working on that. But there's probably at least 3 genes for seizures on that tip. There's a hearing loss gene. Some of your kids have hearing loss, some don't, but we know where the gene sits. That has not been identified yet. There's congenital heart defects. There's actually even more than this. As far as sections go, there's 5 at least. We know some of the genes for sure that cause that. And there's also a cardiomyopathy section. We're going to talk about a gene that causes that too. But do you see how knowing the keyboards of different kids, we can actually find the pieces that are important for different problems, and we can map them. And so this is powerful. Do we still do this? The answer is yes. The smaller your child's keyboard, the more important or the more valuable they are in helping us map genes. And so especially if your child has a very small change, we're very interested in knowing what their issues are so that we can understand the genes that lie in that section. Kids who have really big changes, it's going to be hard because they have so many genes. And so for them, it's not as easy to pick out which genes is causing what. So far, so good. Excellent. Now, going back to that keyboard. So you know that every child can have a keyboard. You know that the keyboard only has so many genes that are really, 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 really important on it. But what we want to do is we want to not only just say that that keyboard, somewhere in that keyboard, is a gene for hypothyroidism or a gene for cardiac or heart defects. We really want to get to the point where we know what that key does. Like, I want to know when you play that key or you don't play that key, which songs get messed up. That would be powerful. Because if I can do that, then I can take any child's keyboard, look at the genes, and I can say, I need to run the following tests because your child may have a risk for having these defects. Does that make sense? Like, that's powerful. But you have to get down to the gene level. How do you do that? I'm going to go back to another circle thing, okay? We saw how genes are important by looking at a bunch of normal individuals and whether they had changes in those genes. What if we did the opposite? What if we took a whole group of people who had medical problems? You name it, autism, developmental delay, cardiac defect, eye defect. What if we took all of them and did the same kind of tests we did for the normal people? At Baylor, we've done a whole bunch of genetic tests over time. We probably have 100,000 test results in our bank. We can go back to that bank and then say, well, what if the child has this problem? Do they have a certain gene? Or reverse, do they have a certain gene that's not working, and do they have the same problems? So for example, let's say I'm interested in this one gene, and I find that 3 out of those 100,000 patients, this is often like how rare these things are, 3 out of 100,000 have a single gene that doesn't work right. Then I can go back and say, I want to talk to every one of their doctors. And I'm going to try to find out if all those patients have the same medical problems. If they do, then just by chance, that would be extremely unlikely. In fact, you can do the statistics on it, and it's like super unlikely that they would have the same medical problems, especially if it's a rare problem. And so we do this, and by doing so, we can identify what each gene does. Let me give you, for instance, do you remember that gene called PRDM16? We looked at it just a little while ago. That was one that we said was a very important, had a PLI score of 1, right? Well, there was a group that took and looked for what is that gene doing. So they found 7 individuals that had that gene not working. And they only had, so they were basically missing 1 copy, or 1 copy was not functioning right of that PRDM16. As it turns out, all of them had cardiomyopathy. Now you may say, what's cardiomyopathy? This is not a hole in the heart. This is that the heart muscle doesn't work right, or doesn't work right over time. It can make the heart get really big. It can make the heart not work right. Some of your kids might have this. But all of them had cardiomyopathy. That's unusual. If you say they also took a look actually at 1p36 kids, and they lined them all up and said, what if the kids have cardiomyopathy, do they miss this gene? Or, in other words, is the gene on every one of their keyboards? And as it turns out, every one of the ones that was in the literature or been described, all of them lined up, and all of them had this gene that was missing, except for 1. This person, or this child, probably has a deletion that goes on over here somewhere, and it probably is deleting another gene that's important for cardiomyopathy. But that's pretty amazing. When you see that, you're like, oh yeah, you have my complete attention, right? How does this help us? Well, first of all, they describe their findings, which is what we always do. We try to send it out so people can learn about this. And they said, hey, if you're missing this gene, you need to be screened for cardiomyopathy. So, how does this help? Right now, we would say that is part of our guideline. In fact, many of your kids probably have been checked for cardiomyopathy. It's probably more important or most important to do so if they're missing this gene. We're trying to figure out if they don't miss this gene, is the risk so low that we don't have to do this anymore? We're not 100% sure yet, but there's more and more evidence that this is the major gene for cardiomyopathy. We would suggest that kids that have this gene missing should be screened for cardiomyopathy about every 2 to 3 years. Or if there's been abnormalities, they should do as much as the cardiologist says. But this is important. So we screen them by an echocardiogram, like an ultrasound for the heart. What does the future hold? There are models, so there are animals that are missing this gene. There's fish that miss this gene, and they have bad hearts. There's mice that miss this gene, they have bad hearts. And so you could say, well, if we could use those mice or fish, could you test something that would make this better or stop that from happening? We would love to do that. That's the kind of stuff that would be really impactful, right? And so that could be a benefit to kids that have 1p36, but it also could be important for kids who don't have that. They don't come to these conferences, but that gene is hurt in them. If we can figure out how to help them, then we can figure out how to help you. And so that's one of the things I think is important, is that we're here because we're 1p36, right? But they could be a whole different conference that's about this gene, and all the kids in that conference are hoping that this gets solved too. So far, so good. Okay. Let's go for another gene. This is one of my favorite genes. It's a gene that I've been working on for about, over a decade. I was sure that it was important in 1p36 because I work on the mice that have this problem or problems with this gene. And they had all the same things that 1p36 kids have. Now you can say, the difference between 47.5 and 3, that's a big difference, right? Yeah. So this is one of those very important PLI score 1 genes. What does it do? So we didn't know for sure what it did. We knew what it did in mice, but we had to wait for the humans to be found. And how did they get found? Same way that we did the other one. We took a look at all these people and said, anybody who's missing RERE, we'd like to know what you have. And so we collected 19 individuals from all over the world that have mutations that affect RERE. We did it through a thing called GMatcher, kind of cool. Can you imagine, okay, you've got a bunch of scientists, a bunch of doctors. We don't have time to do Facebook. We don't do this very much. But at the same time, we recognize that social media is kind of important, right? Well, someone had this great idea. Why don't we just make a database where if you're interested in this gene and I'm interested in this gene, that we get put together? So that thing is called GMatcher. If you go on GMatcher, I can sign into GMatcher and say, hey, today I saw a patient in clinic who has a problem with this gene. I wonder if anybody else out there has a problem with this gene. And if so, do they have the same eye defect as my patient does? I can put that gene into here. It will take my email address and send it to every other person who has an interest in this gene. And I will get the email address from everybody who else is in that. Then I write an email that says, hey, I saw a patient that had an eye defect today. Do you guys have patients that have eye defects? And then they email me and say, yeah, in fact, we think this gene causes that. Do you want to work with us? And this is how we build collections all over the world for different genes. And then we spend the time, it does take time, we spend the time to write this thing up and say, this is what this gene does. So, GMatcher is awesome. We were actually, I was a little skeptical when it first came out, but then again, I got converted after a while because we find so many genes this way. What does this gene do? Remember how we talked about how there's a critical region on the top and a critical region down below? These are kind of all the things that a lot of the kids with 1p36 have. So the developmental delay, intellectual disability, poor growth, brain anomalies, eye vision, blah, 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 all those things. When we looked at the 19 kids that have problems with RERE, RERE is located on that proximal critical region. they have all of these things. And so that one gene, just one gene, can give you all of these medical problems. This is a major, major player. And so we're interested especially in learning how this gene works, because if we could fix that or get around that, that would be really powerful. But again, there are families in groups just like this that are interested in that gene, and they're actually, their interests are also part of what you should be interested in. Isn't that cool? So you thought this was like a big, big group, right? The group is huge. There's a bunch of different groups all over that are interested in this. They just don't quite see it the way you do, because you see it as pieces of your keyboard. They just see it as one key that's not right with their child. So far, so good? Yeah. So we published this out, and there's 2 articles that are out. We also do mouse work with this, so we know how, for example, this gene works in heart. Like we know how it makes heart defects, and we're working to try to figure out how it makes other problems too. What does it look like when we took a look at 1p36 kids? Some of your kids actually are probably in this study. And so if you look, this is the gene. These are all the kids that are deleted for this gene that are in our database. And so, yeah, it's just to realize again that there's a lot of 1p36 kids that are missing this. Not all, but there's a lot. And it could be that your child has this gene that's missing too. How does it help? If I see a child that has 1p36 and they're missing RERE, I'm going to do a super detailed eye exam. Not me. I'm going to give them an ophthalmologist that does that, because their eye defects are sometimes special and could be really important to detect. We're going to do hearing screens on them. Most of your kids are going to get hearing screens when they're born, but I'm going to be especially careful about their hearing. They'll have to get an echocardiogram. They also can have problems with their kidneys. I can't see kidneys when I go and see you in the office, right? So I'm going to get an ultrasound of your kidneys to make sure that those kidneys are working right. Just things that we do, things that we think about. What does the future hold? We have mouse models. We have tons of mice that have this problem, and we are using them to try to figure out how it all works. And again, it could benefit your kids, but it also could benefit all the kids that have RERE mutations. Ready for one more? Okay, there's another gene called SPEN. This is my new favorite. I just got mice that have SPEN in it, or a SPEN gene that's deleted, and I'm going to be studying them in my lab. SPEN has 92.8, big gene, 92.8 people should have this, and only 1. That's a bad gene, right? That's a gene that makes a big difference. And so we were interested in it. We predicted that this gene would cause cardiac problems, because we know that this gene interacts with RERE in the heart. And so actually the 2 of them, when they're deleted together, probably work together to make heart problems. And so we were interested in this gene, but we didn't know what it did in humans. Well, a bunch of scientists and doctors got together on GMatcher, and together we pulled together a bunch of kids that have SPEN problems. It looks like SPEN's important for neurocognitive, so like developmental delay, intellectual disability. We didn't think it would play such a big role there, but it does. And it also, a portion of them also have cardiac defects, which is what we predicted. But again, we're interested in how SPEN works, but it's only because we can put all this together. Do your kids have SPEN changes? It's a little bit farther along on the 1p36, so not a lot of kids have this, but there's still a bunch that have this, and we would say this is probably contributing to the problems for the kids that have this on their keyboard. How is it going to help? Same kind of story, right? We're going to screen these kids by echocardiogram, and they need to have their hearts checked out really well. And again, we have these mouse models that we're hoping are going to help us figure out how to change that, like how to make things better. If you said, well, let's take a look at all the genes on 1p36 that have PLI scores of 1, right? Like the top players. There's actually, all these genes are top players. Now, remember that this is on the entire keyboard, the whole thing. Your child is probably going to have only a subset of these that are going to be, because I don't know anybody, there's very few kids that have the whole thing deleted. Instead, most of us have a small section of the keyboard. But the 3 genes that I talked about are highlighted. Then if you said, well, how many of the other genes are known to cause medical problems? These are the ones in green. So all of them have some type of medical problem that's been associated with them. A lot of them function important in brain development, but there's other ones that may be important in other things. Sometimes I wonder whether it's working the same way in 1p36 as it is in other patients, but this is nuances that we play with. If you said, well, how many of all the genes that are even just PLI scores of 1 are known, by you just doing the math, less than 50% have a actual function that's known. So if you want to think about it that way, we're halfway done on the very, very, very top genes. So there's a whole lot to learn, right? This does not include anything that's less than a 1. Like if it's a 9.5, it's not on this list. But there's a lot of work to be done. But we just can't go at it one at a time. Do I work on all these genes? No. The good thing is somebody else does. And so all these genes are going to be looked at by other groups, and they've already started to make inroads in what they do. So again, I hope you feel slightly empowered by the fact that there's a whole lot of people that actually are interested in you. Even if your doctor doesn't know what this is, even if they're like, what? There are people who do know and care about the keys on that keyboard, and they're trying to figure these things out. So how do we get more stories? In other words, how do you help this? One possibility is you can sign up for research studies. We've had a lot of different research studies that have been going on in different conferences and such, focused on different things. I still run a research study where I work on specific genes, but I'm still interested in learning more about keyboards. In other words, I'd be interested in your child's keyboard, and if it's a big one or a small one. If you're interested in doing that with me, like I'd like to have my child's keyboard looked at, we would be happy to do that. It's pretty easy because remember that one copy of that test report? That's pretty much all you need, is that and then a copy or a description of their medical problems. We put that together in a database, and then we use it to help teach us about genes. And it's actually quite powerful. If you want, you can email me and say, I'd like to do that. If you want to do something simpler, there used to be a day when I didn't used to text. Like literally, I started texting like probably about 3 years ago, 4 years ago, because I was like, I don't want anybody to like constantly hound me, right? But finally, I just gave in. I said, okay, I'll text. And so this is probably even easier. If you have interest in doing this and you want to just text me your name, like even right now, and just say, hey, I'm interested. This is who I am. Text me at my little telephone number and give me a little break because life's really busy right now, but I'll get back to you. If you can give me your email address, that'd be even more powerful. Like text me your email address, and then we'll send you off information on how you can join our study. I think it's kind of fun, and I think it's powerful, and I bet you have kids that could teach us something. Like your kids have already done so, and so if there's people who would like to be part of this, we would love to have you. This is Andres Hernandez. He works in my lab. He's our clinical coordinator person. He speaks Spanish, he speaks English, but he would reach out to you and say, this is how you can join us. But it's pretty easy. You have to fill out a couple forms and then send us your test result. There's also a constant need for more money to do this work. We've seen a shift in that it's harder and harder to get funds for certain projects. And we get that. We're going to be working on this probably forever, whether we're funded highly or not. We always think about this, though. If you are independently wealthy, we would love to have more ability to do this, because our limitations are in part because of how much funds we have in order to do stuff in the lab. Science is actually pretty expensive, just is. And so because of that, again, if you're independently wealthy or have some great desire to do this, it would be a good thing. Even if the money didn't come to our lab, you could pick your keyboard and fund somebody in your keyboard. That would be awesome. I'll just take I think we have some time, right? I I already have my watch is buzzing with all the people who are texting me. That's a good thing. If we have some time, I'd be happy to take some questions until we need to rotate off. Yeah, it'd be nice if we had another microphone. Thanks a bunch, though. I love coming here, talking about this kind of stuff. By the way, I will also be at the question and answer session panel thingy. But if someone has some questions right now, we can talk until Ken says no.
Recently read an article about blockchain being involved in genomics. Have you heard anything about it, and what are your thoughts on it?
I'm sorry, it's blockchain?
Yeah, blockchain, like Bitcoin technology, cryptocurrency being converted to actually study genomic data.
Oh my goodness. Wow, you like blew me out of the water. Like, no clue on that particular one. Sorry. I don't know if that's, no, I know nothing. So my wife will have to read about that and tell me about it is what she'll have to do. Don't know.
Raise your hand high if you.
Is there any way to graft a gene from a brother or sister into a person's genealogy?
Yeah, good question. Actually, we get this a lot. The quick answer is no. And the quick answer is there's not a way to put a gene in to a cell, like not normally. We actually take mice and we can knock genes out. We can put genes back in, and we can do that in mice, but we're actually doing it at the stage where it's just an egg. And so we're doing that very, very early, and we can modify mice that way. The difficulty with here is that your child has not billions, trillions of cells, and every cell has a copy of all the DNA. Even if we could get a new copy of, let's say, a gene into a cell, we can't get into every cell. This is the difficulty. You'd also have to not just get one gene in there, but you'd have to get the whole chunk, like the whole keyboard would have to go in there. That's actually super challenging because the bigger a piece of DNA is, the harder it is to move into a cell. So this becomes very, very difficult. So if you said, well, is gene editing a possibility for our kids? And the quick answer would be no. This is going to probably solve more of the cases to begin with kids that have things like Duchenne muscular dystrophy, where one gene's affected, or like the RERE kids that one gene's affected. But even then, you have to get it to every cell. Super hard. You can get it to liver really easy. And so we're looking at liver diseases that have that and say, can we deliver a new gene there? But this is a very difficult thing. The other thing that people ask me about is stem cell therapy. Stem cell therapy, I don't know, we actually use this on a regular basis. We use it for blood diseases. And so if you have, for example, certain kinds of cancer, or your child does, we can take stem cells and we can actually use that as a treatment. That requires us to destroy the bone marrow that's already there that makes the blood cells. And the good thing is you can do that. You can irradiate a person and destroy all those cells and start them all brand new again with a new batch of cells. We do this all the time. Not me personally, but we do it as doctors. So people say, well, could you do the same thing for 1p36 or some other problem? The difficulty is that you have to replace cells in order to have that work. In blood disorders, you can destroy all the blood-making cells, and then make them brand new. But if people say, well, could you help my child with his brain function? Very simply, we cannot replace all the neurons in the brain. Like, we can't do that. They're already in their place, doing what they're supposed to do as best they can, but I can't change that. And so people say, well, could we do stem cell therapy? Or they'll say, I'm going to go to Mexico or China to do this. Is that a good idea? And just biology-wise, that doesn't make a whole lot of sense because you can't change all of them. Like, you can't replace all of them. If these things would work, we would love it. If at some point in the future we could do that, we're all about it. But for right now, there are certain disorders where stem cells really work, and we do it. And then there's disorders where stem cells don't work, and we don't. And I would say, again, if anybody's thinking about going to a different country to do something, you would probably be better to spend the money on something else, like a communication device or a new wheelchair or something else that would be really helpful, because the chance that that will make a big difference is, if it make a big difference, we do it. We do it here. And so it's challenging and sad, but this is an important thing, because there's a lot of people who would like to make money off of you and your child, and they don't have good things to offer.
I have one more question about that. Okay, if stem cell research has started with embryos and you have your child tested before it's born while it's developing in the womb, can you insert those genes?
Still too late. So yeah, I'm going to give you a more detailed answer, okay? So let's say we want to do this in a mouse. So what we do is we take a bunch of mouse eggs and we do our genetic manipulation, but only a small subset of those eggs will actually we'll actually be able to do it and be successful. Now we're okay because mice are expendable, right? And so we think about it and we say, well, if I can get 1 out of 1,000 mice or 1 out of 100 mice to have the change I'm looking for, I will just have a way to check for that change, and I'll pick the 1 mouse out of 100 that it works. And so even at the embryo, at the egg level, this is hard to do and not very successful, and that we're only getting a subset that actually work. And so once you think about, like, when do you usually get a diagnosis? Like, even if you do it in utero, can you do a diagnosis of 1p36 before a child's born? Absolutely. But we're talking usually about 20 weeks, maybe earlier, maybe 13 weeks if you did it really super early. That child is already a whole bunch of cells, super big. And so changing it even at that level, we are way beyond this. It's just as a kind of, for instance, heart's already formed, brain's well on its way to developing. Like, all these things are already in place. So yeah, when we do mice, we're talking 1 cell right at the beginning. The difficulty also is you say, well, is there a way to get around this some other way earlier? There's no way to test. There's no way less than actually like in vitro fertilization where you can test embryos before they're implanted. But also, this is a very rare disease. And so even if you were to say, well, we're going to have everybody do in vitro fertilization, which would be really expensive and impossible. But even if you did that, we can sometimes only check for one thing in an embryo. And so I can check for a few things maybe, but I can't check for everything. So sometimes parents say, can you guarantee us if we do in vitro fertilization that we can have a normal child? And the answer is no. And they say, why not? Can't you test all the genes? And the answer is no. We really can't do that kind of study even in vitro. And so one of the things you can say is, like, maybe this is just a side thing, but it's important that you hear this. A lot of times parents say, well, is this my fault? The answer is absolutely not. Is there something I could have done? The answer is no. You know, like, this wasn't because of the drink you had or the party you went to or what this happened or your husband's crazy. It's not because of that. This is just chance things that happen. And every child that's born is going to have some chance of this happening. And so no, it's no fault, no anything. Sadly, there's nothing we can do to prevent it. Like, that would be great if we could just do that, right? But the technology and stuff to really be able to do that is not here. So even in vitro, there's no way that we can guarantee a normal pregnancy. Just can't. I like your questions. I wish I could do some of these things. But are they likely to occur in our lifetime? It would take a massive change in technology. Like, we'd have to just have some quantum leap to be able to do some of these things. It might be possible someday, but with the technology we currently have, these are things that cannot be done. Not today.